Well, hello, everyone. And I hope you enjoyed the networking sessions. I know I had a great time in mine. It is now my privilege and honor to announce this year's Cutaneous Lymphoma Catalyst Research Grant awardees. And some of you may not know, we originally launched our Catalyst Award in 2019 as a bridging mechanism to fund research that for already existing projects. And since 2019, this grant has been expanded to also support cutting-edge pilot projects, as well as quality of life research, which you'll hear a little bit about in a minute. So this year we had an incredible response of 22 applications from researchers around the world. A lot of people doing a lot of great work in cutaneous lymphoma. And we really wish we could support them all and fund them all. And hopefully someday we can, maybe with all of your help. But this year, we're thrilled to support 2 Catalyst grants, each for $50,000. So coming up, you'll hear from this year's awardees, Dr. Cosmin Tegla, who's an assistant professor at NYU Grossman School of Medicine, for his pilot project entitled Repurposing, and I will probably butcher this, Adovanquan, an FDA-approved antimicrobial for the treatment of cutaneous lymphoma. Whoosh, that's a mouthful. And Dr. Neha Akkad and her co-principal investigator, Dr. Neha Mehta Shah, both of Barnes Jewish Hospital in St. Louis, for their quality of life project entitled Quality of Life in Patients with Cutaneous T-Cell Lymphoma, Validation of a Novel Quality of Life Instrument. So congratulations to both of these amazing projects and researchers. We're very excited for the work that they're doing and grateful for their work that they're continuing to move research forward in the area of cutaneous lymphoma. So now I'll let them tell you a little bit more about each of their projects.
Hello all, my name is Neha Akkad. I'm an internal medicine resident training at Washington University in St. Louis, and I'm so grateful to the Cutaneous Lymphoma Foundation for selecting this proposal for the Catalyst Research Grant, and I'm so excited to tell you all a little bit about this project. This project is entitled Quality of Life in Patients with Cutaneous T-Cell Lymphoma, Validation of a Novel Quality of Life Instrument. The overarching aim of this project is to create a scientific tool in the form of a survey that is both comprehensive and disease-specific to evaluate quality of life in patients with cutaneous lymphoma. As many of you may know, and as we have seen in previous studies, cutaneous lymphoma has a negative impact on patient quality of life, even when compared to other similar skin diseases and cancers. This is due in part to the fact that cutaneous lymphoma has unique symptoms, treatments, and psychosocial burden on those affected. However, thus far, a tool to properly evaluate quality of life in all stages of cutaneous lymphoma patients does not exist. We've therefore set out to create a new tool that fully encompasses all themes important to cutaneous lymphoma patients that will be made using the feedback from our patients. We have so far conducted interviews with cutaneous lymphoma patients being treated at Washington University in St. Louis and analyzed what themes are most important to their quality of life. From this data, we will create a new survey, which we plan to distribute along with several previously existing surveys in order to analyze its performance. Eventually, we hope that this survey can be used as an additional tool to properly evaluate and thereby address quality of life in patients with cutaneous lymphoma. We also hope this tool can be used as a metric in clinical trials to specifically correlate quality of life with treatment and response to treatment. I am so thankful for the support of the Cutaneous Lymphoma Foundation for this project. As a young clinical investigator, I feel incredibly honored to be able to work with our patients to better understand how to improve their quality of life. It's truly a privilege to be able to further this research. Additionally, having the ability to include the voice of our patients in the creation of this survey is truly special and will hopefully lead to a meaningful way to evaluate quality of life that can be used universally. I also hope this project will highlight the importance of quality of life in this patient population. In closing, I would like to extend a huge thank you to my mentors, Dr. Neha Mehta Shah, Dr. Amy Musiak, and Dr. Donna Jeffy. Most importantly, a huge thank you to the Cutaneous Lymphoma Foundation for this incredible opportunity. I look forward to sharing the results of this study with you all in the future.
Hello, my name is Cosmin Tegla. I'm assistant professor at NYU Grossman School of Medicine, working in the Department of Medicine, Division of Hematology and Oncology at NYU Langone Health. My project, named Repurposing Atovaquone for the Treatment of CTCL, is aimed at investigating the therapeutic potential of atovaquone as a novel agent for the treatment of CTCL. Atovaquone is an FDA-approved antimicrobial being used in the clinic for over 20 years for the treatment of or prevention of malaria, PCP pneumonia, toxoplasmosis, or babesiosis. And it is a safe drug with a good track record. Our preliminary data show that atovaquone effectively blocks the proliferation of malignant CTCL cells and specifically inhibits the viability of the malignant CTCL cells with little impact on the normal T-cell counterparts. The hypothesis of this study is that atovaquone blocks specific vulnerability of the metabolically rewired malignant cells. And the aims of my projects are to define the metabolic changes that are induced by atovaquone treatment to help determine what this underlying vulnerability is. And second, to determine the mechanism of action of atovaquone by using a genome-wide CRISPR-Cas screen aimed at identifying key effector genes. Next month, I'm joining our faculty group practice at NYU with the aim of becoming a T-cell lymphoma expert and develop into a translational researcher. My ultimate goal is to bring solutions from bench to bedside, and conversely, to investigate relevant clinical questions with the highest granularity that is possible by using top-notch basic science tools. This award is a big step towards achieving that goal, and the timing couldn't be better. It is actually perfect. And I feel honored to receive this award. In short term, atovaquone could become a new therapeutic option for the treatment of CTCL because of its current FDA-approved status. Because it's a safe drug, it could be easily benefit patients with early-stage disease where the goal treatment is to minimize interventions while maximizing quality of life. And at the same time, it could benefit patients with advanced stage disease where the goal of treatment is to maximize responses and duration of remissions. In either situation, atovaquone could be easily combined with the standard of care to help improve outcomes with the minimal safety concerns. And in long term, atovaquone could help identify, this project could help identify novel therapeutic targets in CTCL and perhaps other malignancies that share similar biology. Based on our preliminary findings, atovaquone targets CTCL in a novel and unique way, and elucidating its mechanism of action could lead to new therapeutic strategies in CTCLs and potentially have a broader impact on other malignancies that have similar biologies. So I feel honored to receive this award, especially at this moment. And thank you. I would like to thank the Cutaneous Lymphoma Foundation for such an opportunity.
Thank you, everybody. And we'll have a few minutes in between our next sessions, which will start in 5 minutes at 2:45. So you get to pick one of the breakout sessions. We have 3 to choose from: the scientific approach to understanding cutaneous lymphoma, quality of life, the long-term impact of living with cutaneous lymphoma, and the ever favorite, itch, itch, itch. So we'll see you all here in another few minutes. Thank you, everybody. And congratulations to our Catalyst Award winners again for this year.